Now we will talk very briefly about the differences and similarities between the prokaryotic and eukaryotic DNA replication. First of all, they are pretty similar. They use the same basic process, same basic requirements. You need enzymes to bind the DNA, you need enzymes to unwind the DNA, you need enzymes, the DNA polymerases to add nucleotides on, so you need raw nucleotides, so same basic components really. However, there are differences, and the main difference is because the eukaryotic DNA is just simply much more complex. And it's more complex because we have a lot more DNA, and the DNA is going to be complex with nucleosomes. And we'll talk more about the structure of the chromosomes in the next couple of lectures, and so that'll be chapter 12 material. The other major difference is that the eukaryotic DNA is linear. And so what we will see, if you look on the left here, you can see the typical bacterial or prokaryotic replication. Again, starts with the origin of replication, it's bidirectional, and after it reaches the termination region, the replication is complete. In eukaryotic DNA replication, we have many, many different replication forks or bubbles that happen at the same time. So within these bubbles, there are actually two replication forks, and they're occurring bidirectionally. As replication proceeds, these replication forks become nearer and nearer to one another until they become basically one long complete strand. And at the end of it, you have the two new DNA complexes. We do this because there are several different reasons. First of all, they're much larger, they're much longer, and the polymerases just aren't as fast. So if you were trying to do replication and you tried to do it in the same exact manner as the prokaryotic cells, it would take days, years maybe, I don't know, to replicate a single cell. This way they can be much more efficient at trying to do their replication within a timely manner. So these mammalian genomes, they may contain as many as 25,000 replication origins. And compare that to the one that we find in typical prokaryotic cells. There are also many different types of DNA polymerase molecules and many different numbers of them. So maybe 15 copies of DNA polymerase per cell in E. coli. Mammalian cells may have tens of thousands of individual DNA polymerase molecules. And again, if you think about these 25,000 replication origins, if all of them are active, you're going to need a huge number of polymerase molecules to keep up with that. There are also many more types of the DNA polymerase than in prokaryotes. Remember, in prokaryotes there were 5 types, and in eukaryotes there's at least 14 that we know of. Just briefly, we're going to talk about chromatin. We're going to talk about this in more detail in just another lecture or two. But the eukaryotic DNA is complex with DNA binding proteins, and we call these nucleosomes. Every 200 base pairs of DNA are complexed with 8 histone protein molecules. Histones are just proteins. And what we find is that they are very prevalent. They disassemble right before replication, and they reassemble right behind the replication fork. They do so through other proteins that help this process called chromatin assembly factors. So because they're pretty much always there, they're only removed really when needed, it's indicating that they really are forming a real important function.